Okay, so now we're going to take a look at some real-life use cases of where FunCoup has been used. And then I'm going to show you how you can use the website, the FunCoup website, to analyze and further investigate genes and their interaction partners. So to start off, I'm going to tell you a bit about this study. It was released in 2016, and it's an article on the sigma-1 receptor by Su et al. The article explores the sigma-1 gene and describes its various functions and involvement in numerous diseases like Alzheimer's disease and cocaine addiction. They conclude that the sigma-1 protein class could be called pluripotent modulator due to its many functions. Let's see if I can. Oh. Okay. So in their study, Su et al. Looked at 19 experimentally verified interaction partners to the sigma-1 receptor and compared them to 29 predicted interaction partners from FunCoup 3. They found a quite low overlap between the two sets, where only two genes, HSPA5 and BDAC, overlapped. And they sort of left the analysis there and concluded that many of the high potential interactors based on the FunCoup predictions have not yet been tested in, for example, proximity assessment assays. So taking a closer look at the sigma-1 gene, we can find in UniProt that it's an endoplasmic reticulum protein, which is involved in, has a lot of functions. So among others, involved in lipid transport, regulation of biogenesis of lipid domains, regulation of receptors, regulation of ion channels, and calcium signaling. So now it's time to take a look at the interaction partners of the sigma-1 gene in FunCoup. So let's see if I can make this work here. Yeah. So this is the FunCoup website. This is what it looks like. So we can start by typing in the sigma-1 gene here in the search field, and then just press search. Eric is later going to show you the advanced search and other settings that you can do before searching if you want other or more specified search results. So here is the subnetwork that we get for the sigma-1 gene. As we can see, the sigma-1 gene is in the middle. In this subnetwork, we can also find a few genes that were predicted by the Su et al. Study, the ITPR3, BDAC2, and BDAC1 gene here. So in this view, we can also try adding some tissue filtering. So since we know from the Su et al. Study that the sigma-1 gene is involved in various central nervous system related diseases, we could filter this by cerebral cortex. And by doing so, we can see that a large section of the subnetwork remains intact. So moving on to this interactions tab, here we can see a list of all of the interaction partners to the sigma-1 gene. So by default, it will list the top 30 interaction partners. Here we can see that all of them has quite a high confidence score, above 0.9. And we can also see which gold standard networks the highest scoring interaction originates from. So here we find a lot of metabolic and a lot of PPI for the various links. Then we can take a look at this evidence types matrix, which will show you the score contribution from each evidence type to the existing of the link. So here we can see that a lot of evidence is derived from MEX or mRNA co-expression, and also from PIN, protein interaction, and SCL, subcellular co-localization, by these green squares here. Then here right next to it, we have the species matrix, which shows which species orthology transfer of the evidence has been derived from. So as we can see, or as we might expect, like a lot of them actually derived from Homo sapiens or human. But we can also find some S-cerevisiae evidence transfer here with these green boxes. Then we can also dig deeper into the evidence actually supporting a given link. So if we take a look at this MSM01 gene, that was not expected by the Su et al. Study to be an interaction partner to sigma-1. So you just expand this by clicking this little eye symbol here. So this gives us a list of all of the evidence supporting the link. So here we can see that these genes are co-regulated by a number of regulators from SRVCA. We can also see that they have a high correlation in a number of mRNA co-expression data sets, and also in SRVCA. And we can also see that they have shared cellular co-localization in human. Next, we're going to take a look at the next tab up here, the Interactors tab. So here we can find some more information about our genes that we're interested in. So if we expand here, we can see a number of alternative identifiers representing the genes. If we take a closer look at this MSM01 gene that we saw earlier, we can also see that this gene is actually a part of the KEGG metabolic pathway steroid biosynthesis. While we can see that the sigma-1 gene is not actually part of a KEGG metabolic or signaling pathway. So from this view, we can also go to the enrichment of this gene set in PathPix. So by just clicking on this button here. So I will show you the results of this. So here we get a list of significantly enriched pathways. And let me just click some filters here. And maybe zoom out a bit so you can see all of it. So this is the list of enriched pathway for this subnetwork that we saw around the sigma-1 gene. And as you can see here, you may recognize this pathway, the steroid biosynthesis pathway, which we know that the MSM01 gene is actually a part of. The sigma-1 gene is not part of this pathway, but it does have a lot of links to the pathway genes, so it has a strong crosstalk to those. And also when we look at the GA overlap, we can see that this is the only pathway that we would have found if we would have used GA for pathway enrichment. So none of the others would actually have been detected. But by using PathPix, we can see that we have a list where we have a lot of fat-related pathways, which we could sort of expect, as we know from UniProt, that the sigma-1 gene is a lipid transport, involved in lipid transport. And we can also find the pathway cocaine addiction here, which, as we know from the Suedel study, the sigma-1 gene is known to be involved in. So that's all I had. So to conclude, I've just given you a simple analysis that you could do around the gene set and its interaction partners.